With the construction of the new small animal imaging building for the Bioimaging Lab and the Molecular Imaging Centre Antwerp, MICA, the University of Antwerp brings preclinical imaging to a whole new level. Molecular imaging, both nuclear and MR, has become increasingly important recently and forms a key factor in innovative, fundamental research on diseases like cancer, Parkinson's and Alzheimer. The Bioimaging Lab uses both magnetic resonance imaging and bioluminescence imaging and has always played a leading role in preclinical brain research in mice and songbirds. The lab now has 3 MR scanners for small animals next to a BLI scanner. Next to the Bioimaging Lab is the new Nuclear Imaging Lab, which makes it now possible to do PET scans as well. The Nuclear Imaging Lab uses molecules that have been made radioactive. These molecules are injected into the animal, which is then put asleep. By using the new state-of-the-art scanners, the molecule can be followed throughout the animal's body. The fact that both the Nuclear Imaging Lab and the Bioimaging Lab are now so closely linked together and literally next to each other is a big advantage for both labs, since both imaging techniques are very complementary to each other.
What is most advantageous is that we can study the same animal almost consecutively with imaging tools we have, and then move with the animal in the animal bed anesthetized to the other department and then investigate it with the tools they have. So you can apply an entire sequence of imaging procedures to the same animal because we're so close to each other. It's only one door.
Both preclinical departments are also closely linked to their clinical counterparts. In fact, the scanners they use are miniature copies of the scanners used in clinical imaging. This allows research to be conducted from A to Z within the walls of MICA.